let's wait a few seconds so everyone can join. So welcome everyone. Greetings everyone. Welcome again after a long break of the EMN live webinars from the Metabolomics Society. My name is Juan José Oropeza Valdez. I'm from the National Autonomous University of Mexico, and together with my colleague Marina Botana, who's in the Karolinska Institute in Sweden, Karolinska, sorry, who's also a postdoc in the same institute. We are really pleased to be your host for today's webinar. I hope you enjoyed today's session. But before we present today's speaker, I would like to encourage you to follow us in all our social media platforms, on Blue Sky, on Twitter, X, and LinkedIn, or send us your questions via email. And encourage you to join the EMN in the following years if you are an early career researcher or a student who does metabolomics. We do all kinds of activities like this webinar, as well as other meaningful activities like the workshop in the Metabolomics Society conference and much more. With that, it is my great pleasure to leave you with Marina, who's introducing properly today's speaker, Dr. Yamilé López.
Welcome everyone. Today I'm pleased to co-host session 21 with Dr. Yamilé López Hernández. She's a research associate at the Metabolomics Innovation Center at the University of Alberta at the Wishards Lab in Canada, and a member of the board directors of the Metabolomics Association of North America, MANA. She also leads the Clinical Metabolomics and Proteomics Laboratories at the Autonomous University of Zacatecas in Mexico. And she earned her PhD in health science from the University of Medical Sciences in Havana in Cuba back in 2011. And her work has significantly contributed to the development of high-throughput metabolomics platforms and diagnostic tools, aiming to improve disease detection and patient outcomes. Dr. López Hernández is the organizer of the annual Making Metabolomics Matter workshop series, held in different Latin American countries to promote the integration of metabolomics into regional research and healthcare. She is also a member of the work group for Interbio 21st, an international initiative aimed at improving the phenotypic characterization of intrauterine growth restriction, small for gestation age, and preterm birth syndrome through the integration of molecular, biochemical, and clinical data. Through her career, Dr. López Hernández has published over 60 scientific articles, patents, and innovations, and mentored numerous of graduate students. So today is a big pleasure to have her at the webinar today, and I'll let her introduce her talk.
So I'm gonna stop sharing my screen.
Thank you so much, Marina. Hi, everyone. Can you see my screen?
Yes.
Okay. It's a great pleasure for me to be here in this webinar with you. I will share some of my experiences, my point of views about clinical metabolomics. More than presenting most of my results, I would like to discuss and to present some of the current challenges in the transition from biomarkers from the bench to the clinical practice. Over the few slides, we will deep into how metabolomics can help us better understand disease mechanism, improve diagnosis, and of course, saving lives. Okay. The webinar structure will be as follows. The first part, I will introduce why we need clinical biomarkers. In the second part, I will be talking about why most of the biomarkers die in their way to clinical adoption and some solutions to prevent this. And in the third and last part, what was done for some biomarkers to survive this pipeline and some general lessons. While not an expert in clinical regulations in absolute, it's better to know what you are going to find in your way to get biomarkers into clinical applications. And this is the purpose of this webinar. There are so many reasons why we do metabolomics, so many reasons. But however, I will focus on three applications, three approaches which will support the objectives of this webinar. First, to build reference ranges for large populations of apparently healthy individuals. And this is to establish normal and abnormal reference values, which allows to predict the early detection of diseases. Currently, there are so many repositories for metabolomics data. However, one of the most famous and most comprehensive is Human Metabolome Databases, or HMDB, containing more than 200,000 metabolites, millions of mass spec and NMR spectra, and covering data for almost 600 diseases. And it's currently updating, and from time to time, every 2 to 5 years, there is new releases with new characteristics and new improvements. The other resource that is very valuable in clinical metabolomics is marker DB. For marker DB, we published the second update this year in 2025. And you can find in this repository information about several molecules, a huge collection of molecules. You can search by compounds, by clinical condition, by type of biomarkers, because we included genetic biomarkers, risk biomarkers, prognostic biomarkers, exposure biomarkers, and you all also will find information, general information about each molecule, but also specific data about the performance, the test, raw curves, the age, sex, and reference values for each of the studies published and included in this database. So you can explore it, and for sure you can use this resource for clinical metabolomics. The second approach we do is to discover metabolomic signatures of disease susceptibility and to see how metabolite changes appear even years before the diagnosis, and of course to describe metabotypes. And there are a lot of papers in the literature showing the relationship between metabolites and diseases outcomes. I was invited last year to participate in this large cohort study with the EPIC. EPIC is one of the largest cohort studies in the world, based in European countries. And in this study, we analyzed the association between metabolites and different status of prostate cancer. The sample size was 851 for cases and 851 for healthy controls, matched perfectly with several parameters. And what we found was positive and negative associations with prostate cancer. And most of the significant metabolites were coming from foods, from vegetables, but also from coffee, for tea, some endogenous metabolites, some metabolites, for example, 2-methylamine oxide, N-oxide, which is also related with microbiota composition and some foods. So we, with this study, found for the first time reports for some metabolites that were never reported in prostate cancer. And it was done with target metabolomics method available in the laboratory of Dr. Cristina Andres La Cueva in Barcelona, in Spain. If you are located in Europe or in Spain, this is a fantastic lab with validated methods, target methods, not only for endogenous compounds, but also for exposomic compounds. With this paper and systematic review we also published, we were able to extract information and to elaborate some panels for prostate cancer. For example, this panel composed by 18 metabolites that we are currently using in the Metabolomics Innovation Center here in Canada, in Edmonton, to as a service, just to evaluate the association and to get more data and more validation about these metabolites and the outcome of prostate cancer. The third approach, why we do metabolomic and clinical metabolomics, is more related to personalized medicine, and is to estimate personal susceptibility, to understand risk trajectories, and of course, to support N-of-1 precision medicine for moving to P5 medicine. I mean, predictive, preventive, personalized, participatory, and psychocognitive. And this is because individuals are incredibly stable over time. Our personal metabolome is more unique than our fingerprint. And when we detect small variations in comparison with our baseline, these could be powerful predictors of diseases, dysfunctions, or also responses to stress and treatments. And let's put one clear example. This case report was published last year in 2024, and it's about one Canadian woman, Diane Ballon, who was using metabolomics and proteomic tests developed by Molecular U, is a company based here in Canada, to consistently measure her blood. And what she noticed is that from 2028 to 2021-22, there was a drastic and dramatic increase in several metabolites and proteins related with some processes that take place in cancer and other inflammatory diseases. And what happened was that her family doctor, who was really confident with our methods, with metabolomics, with proteomics, he ordered the MRI, and they found a pancreatic tumor. Then in 2023, she was operated, and the analysis done in this year reported that most of the metabolites and proteins returned to the patient levels in 2018. This is Diane Ballon, and now she is sharing her experiences with different audience to help promoting these technologies for everyone at any moment, because she was essentially saved by metabolomics and proteomics. And on a more personal note, I want to share a personal story from my life. My daughter, at a young age, she was diagnosed with polycystic ovarian syndrome. And since this moment, we start monitoring her metabolome with a panel of 144 metabolites, a target panel. And we compare because she also participates in this study on the data analysis. We compare the changes before or at the moment of the diagnosis and insulin resistance, and after diet and exercises. And it's fantastic, it's amazing to see how the metabolites are progressively normalized with the time, and every time she maintains the same and regular habits, these metabolites are stable, and we can track different alarm or different changes that predict outcomes in polycystic ovarian syndrome. Having said that, we obviously need biomarkers to detect, to diagnose, and to predict diseases. In this way, we can move beyond one-size-fits-all medicine to get more target and effective treatments. But biomarkers or biomarkers are not just unknown molecules that we discover by metabolomics. Many of them are metabolites which are currently in clinical practice. The key lies in combining these known or unknown molecules in other ways by quantitatively measuring their levels and, importantly, their radius, and understanding how they interact. We can gain insights into individual health. It's just about using some new and existing knowledge to improve the predictive power of our tests and to make them more relevant for clinical practice. Because metabolomics data is enormously rich, but is also enormously noisy, meaning that while we get a lot of information, we also be encountering many confounding factors. For a biomarker to be truly useful in the clinic, it needs to be analytically valid, meaning that we can measure it reliably and reproducibly. It needs to be clinically valid with a strong independent association with disease and health outcomes. And it must have a clinical utility. It should actually change clinical decisions. And in this regard, target metabolomics methods offer the right solution. It offers a clear path toward clinical integration and translation. By quantifying these metabolites, we will simplify the process of establishing cutoff values, preference ranges, making them more suitable for regulatory approval, which is what we need at this moment. And I'm not saying that on-target is not valuable, but if we see this slide, in 2010, there was one paper reporting that the identification of compounds from mass spectrometry data is still seen as a major bottleneck. It was in 2010. And then yesterday I came across with this paper published by some of our colleagues, for example, Armando Alcázar, who is in BC, in Vancouver. And they published a nice paper reporting that there is still so much to do in on-target metabolomics, despite the outstanding progress that the quality control, quality assurance and consortium it's doing for on-target metabolomics. There is still the bottleneck in identification of compounds and the assignment of identities to several compounds, confounding in some cases with blank or some other components. So there is still, 15 years later, there is still the bottleneck in on-target metabolomics. And I say this because I'm coming from the on-target world. And I initially performed some on-target studies in 2019. We published these results for gestational diabetes mellitus. And then later in 2021, we used the same samples to validate by target our hypothesis in on-target metabolomics. And also we found some metabolites coincident. Not all the results were able to be reproduced. For example, the kynurenine tryptophan pathway was not found in target methods. In another paper we published with on-target analysis for long COVID patients, we reported several lipids, phospholipids, lysophospholipid that were downregulated in long COVID patients. By fortune, these lipids were validated in the target assay in 2022, and it was also published. So I mean that even when we are doing everything correct for on target, there is always, it's not possible to reproduce or to validate the results, because we have so many influence about the instrument status and some other interference that we need to work more together to solve. But also, even doing target metabolomics, most of the potential biomarkers are on their way to clinic. One reason is overfitting in discovery studies, where models trained on small data sets give optimistic results that don't help up on larger independent cohorts. And in this case, it could be probably useful or benefit to implement mixed hybrid strategies for machine learning. For example, those based on genetic algorithm. This is one thing that Dr. Juan José Oropeza and myself are trying to implement in our last papers to compensate a little for small sizes and nonlinear data, and also highly correlated metabolites. Another reason of failure are the pre-analytical factors that can lead to erroneous results. I can recommend this paper. It's an excellent overview and summary of all the pre-analytical factors that could happen in the metabolomics, and how to implement strategies to prevent these pre-analytical factors impacting negatively the metabolomic results. I will show now some examples collected in my lab during several years. Some of them were accidents, some of them were human mistakes, but we track all these pre-analytical factors just to learn more about what happened. And for example, this was one study conducted in 2019. And the original plan was to do on-target methods with QTOF. But the problem is that during the collection of samples, and the samples were the urine samples from babies, newborns, in the 24 hours, the first 24 hours after delivery. But the nurses put some lotions, some skin lotions and creams in the babies. So the samples were contaminated with polymers, and that's why it caused ion suppression, and our on-target experiments failed. The other example, for example, is how centrifugation delays can affect the levels of some metabolites, for example, glutamine and glutamate. And we realized it because we used reference materials, in this case, NIST. These are the right values for NIST for glutamine and glutamate. And then we realized that in our samples, the ratio was totally opposite. So it means that something happened during the centrifugation process, and the reaction occurred just to produce more glutamine in cells. And then there is another example. It happened in my lab accidentally in 2023. Some samples were left at room temperature overnight, and a part of the drama that it provokes on me, we saw the drastic effect of this accident in the lipid profile, because most of the phosphatidylcholines were hydrolyzed and produced lysophosphatidylcholines. Of course, these samples could never be used anymore in any experiment forward as a Greenland or England. And the other example, for example, is this one. Some researchers mixed serum and plasma samples, but they wrongly labeled the samples as the same biofluid. They said, these are serum samples, but not. When we analyzed it, and we thought that we were having sarcosine as a putative biomarker, we noticed a drastic increase in the concentration of sarcosine in one entire group and in comparison with the other group. And what we saw later in one paper we recently published is that sarcosine is one of the metabolites that is differential in venous plasma respect serum plasma. I mean, this is a marker that could help you to differentiate between plasma and serum. And in one case, you have a question about if your samples are right or your samples belong to serum or plasma. You can use sarcosine to identify these labels, because it's a dramatic change. But all of these errors happen in the lab, sometimes caused by insufficient training, some caused by instrument status, but this is because in some labs, and most of the labs, probably we have poor quality management. And this is one thing we need to work with if you want to have success in clinical metabolomics and in the approval of biomarkers. We know that assays have to give always the same answer for the same sample in different instruments. And there are some conditions that you can notice here, and the explanation for what happened. For example, if different people are running the same assay and get different results, it indicates problems in the protocol or training problems. But if the same people are running the same assay on different instruments and get different results, it could indicate instead instrument problems. And if some people report assay failures of about 50% and others report no assay failures, it indicates for sure training or competence problems. How to fix these problems? Basically implementing a quality management system, having a tracking of inventory, the purity of the reagents, developing standard operating procedures, SOPs, that are constantly checked, rechecked, validated, and constantly updated. So this is the way for clinical metabolomics lab. And also to adopt some clinical standards, for example, ISO 9001, ISO 17025, ISO 15189. In specific, this latter ISO It's a comprehensive standard designed to enhance the quality management systems of medical and clinical laboratories. And it means that all the laboratories having this ISO are formally recognized to be competent and approved for medical and clinical analysis services. I list here some of the reporting criteria required by FDA to have a biomarker approved. For example, reference standards, data, calibration curve, critical reagents used in the method, the quality controls, and also some other parameters coming from the validation of the methods. But importantly, FDA expects biomarkers to be absolutely quantified. And now I will show the examples of some successful biomarkers, some others that are in the way for adoption, and some solutions we have found to make the process of approval easier. Our group has been collaborating since 2020 with the Metabolomics Innovation Center in Canada, and we have measured hundreds of metabolites by quantitative methods, using panels of metabolites containing as low as 144 metabolites or 1,700 metabolites, which is a huge amount. We included in all of these studies pregnant women, newborns, patients with different diseases, for example, Parkinson's disease, diabetic nephropathy, cervical cancer. Some of these studies are cross-sectional studies, some others are longitudinal studies. All of them have been pilot studies with a relatively small sample size, but it allowed us to collect relevant data about metabolomics, about different ages, sexes, different populations, and clinical conditions. Since they were not on target methods, they were target methods, but comprehensive methods. Comprehensive because we included as much of metabolites as possible. And this is important for the discovery of biomarkers because biology is interconnected. And in this way, we capture global physiology, not isolated reactions. And we were able to link molecular fluxes to functional outcomes, and more importantly, to determine ratios of metabolites, which it's really important in the validation of biomarkers. This is one example reflecting how one of our assays looks like. It was done by Dr. Juan José Oropeza, just representing some of the molecules included in one comprehensive assay. And we can see that if not cover all the metabolism, we can cover a high part of the metabolism. And for sure, we will make associations about the interaction of the metabolites and how these metabolites could be included in one panel or not to predict different diseases. Our work with maternal newborn binomium allowed us to publish at least 4 papers. And from these papers, we extract relevant data for including those metabolites in the interview 21st century study to prevent restriction growth during the pregnancy and to supplement pregnant women with the right supplements to guarantee in certain mode successful outcomes. But also, we have discovered some biomarkers, for example, trans-hydroxyproline, which is very, very associated with bronchopulmonary dysplasia. And bronchopulmonary dysplasia can be diagnosed early after the birth, probably in the first hours, because bronchopulmonary dysplasia occurs in infants born extremely preterm, less than 28 weeks of gestational age, and the incidence could be up to 89%. So now we are preparing a large study to validate the trans-hydroxyproline in this population, because we have a simple and rapid sensitive quantification through mass spec, but also there are some colorimetric methods available to measure trans-hydroxyproline, which probably could help in the transition of the biomarker to clinical settings. In our work with COVID-19, we analyzed it from 2020 to 2024, different populations in Mexico. And what was possible to do was to know how to supplement long COVID patients with the right compounds, for example, antioxidants and some amino acids, to overcome the symptoms caused by long COVID, for example, fatigue and articular pain, and also joint pain, and insomnia, and some neuropsychiatric symptoms that we found in our population. The most important lesson is that to have all the background about those metabolites, we need to quantify, and quantify. And this is better when we can compare results in different populations, in different settings, with different instruments, in different laboratories, because we are using protocols, we are using harmonization, and we are using quantitative tests with internal standards, with calibration curve that could allow reliable measurements of metabolites. This is another example we did this year with cervical cancer using GIGA kit. And since this kit included not only indigenous compounds, but also exogenous coming from diet and exposome, we discovered some metabolites that were not previously reported in the literature. And now, again, we are preparing larger cohorts to validate the results. Collectively, these studies, when we extract information about all of them, we saw different metabolites. All are non-metabolites. Some of them are practically measured in all these clinical settings, for example, lactic acid, glucose, uric acid. Some of them are carnitine, acylcarnitines, which are used in the newborn screening. But the common thing is that all these metabolites have particular roles in inflammations. And we know that inflammation is the basis of most of the diseases and autoimmune diseases. Now we can see in this slide some of the metabolites that are successful, and they are FDA approved and CLIA regulated clinical assays in most of the laboratories in the world. There are some others that are included in the newborn screening, for example, amino acids and carnitines, acylcarnitines, and some steroid hormones. And there are some other metabolites that we need to pay attention, because although they are not approved, they are near to get some guidelines, and they are used in most of the major cancer centers. For example, some oncometabolites, succinate, fumarate, hydroxyglutarate, but also those metabolites related with kynurenine and kynurenine tryptophan pathway. Of course, some uremic toxins, and we evaluated recently in our most recent paper published in Metabolomics. We evaluated a set of uremic toxins related with diabetic nephropathy, and indoxyl sulfate and palacresyl sulfate are among those metabolites. And for sure, they are totally valuable to evaluate chronic kidney disease progression and also some other cardiovascular issues. Now let's look at what has worked and what we have learned about doing this rigorously. And we can mention in the first place that the winner and the gold standard, the perfect example for clinical metabolomics is the newborn screening. Why? Because there is a strong mechanistic link and a clear path between biochemistry, the metabolite, and the actionable clinical decision. When this happens and one enzyme is blocked and we have accumulation of the metabolite associated with this pathway, we can have a good test just to do actionable clinical decisions. It happens in fatty acid oxidation disorders because every defect in one enzyme or transporter will lead to the accumulation of specific acyl-CoA intermediates. The advantage of this neonatal screening and what has made the winner in the clinical metabolomic is, as I mentioned, the strong biochemical mechanism, but also that mass spectrometry is the perfect technology. Also, the excellent analytical precision and reproducibility. We can mention that there is a minimal influence from diet, microbiome, or environment in the acylcarnitine levels. It could influence the levels, but not to the pathophysiological levels that are observed in the genetic diseases affecting the inborn errors of metabolism. We can also mention that it has been decisive, the dried blood spot that has made logistics easy, and that has allowed the testing of more than 40 million babies yearly worldwide. So we have here all the criteria to get success: analytical performance, a major impact, large studies conducted, some stable tests, for example, dried blood spots, and minimal influence of confounding. Infants identified early can about coma, sudden death, and of course, this is the clearest example of metabolomics changing patient outcomes, as I mentioned with the case of Diane Ballon at the beginning of the webinar. The other story is not like a successful story like a newborn screening, but it's a huge cumulative evidence about the enrollment of trimethylamine oxide in different conditions, for example, cardiovascular diseases. Trimethylamine oxide was discovered initially by on-target metabolomics, and there were so many validation studies proving the presence and the alteration of trimethylamine oxide in these patients. But since 2011 to 2023, more than 10 years, there is still no guidelines to or any clear labs or any FDA approval to trimethylamine N-oxide, because there is no treatment trials using trimethylamine N-oxide as a formal therapeutic target. So although there is so many evidence about the enrollment of trimethylamine N-oxide in cardiovascular disease and hypertension, there is still no approval, at least for this biomarker, formally in most of the labs. And there is another story, for example, Polyp DX. This is one innovative panel that was initially discovered in from 2005 to 2013 through NMR in Edmonton, Alberta. And the panel consisted of 12 metabolites: succinic acid, ascorbic acid, carnitine, creatine, citric acid, fumaric acid, trigonelline. We can see here some of the metabolites that we have consistently mentioned in the previous slides. So it means that those metabolites are common in different conditions and of course in inflammatory conditions that we all know that are related with the outcome and progress of cancer. This test was validated and was translated to MS, mass spectrometry assay. Why? Because NMR is not practical for clinical metabolomics. So we shall note in TIMIC, this assay was modified to include only 3 metabolites: succinic acid, ascorbic acid, carnitine, and in combination with some clinical characteristic and demographic characteristic like smoking status, age, and sex, the test was developed and patented and validated in China and in Canada. And the test demonstrate good performance, even really better than the fecal test available for testing colorectal cancer. So it was validated in 2 populations. The performance were, at least in these studies, was good, in some cases excellent, and it was related with the screening of cancer, to get the screening more easy and to avoid colonoscopy. However, after years of use and use, Polyp DX is not FDA approved. It's only available as a laboratory developed test through some CLIA certified laboratories. And it happened because there were no larger scale studies to demonstrate a real clinic utility. There were some clinical tests that were planned in 2023. They were suspended. And now there are some ongoing tests in Nigeria and some other low or middle income countries, just to try to see if the test could have some other approvals. But if it's restricted to these laboratory developed tests, it will not be widely used in This is one of the examples of the clinical trials that are now taking care in Nigeria, Africa, and some other countries. And there are some other examples we are working with to try to develop a testing test for at-home lung cancer screening. This is a collaborative work between University of Alberta and BC Cancer in British Columbia. And in this case, again, the panel of metabolites was validated in 2 different populations in China, in Canada, with samples for more or less 1,000 samples together in both populations. This is a panel consisting of 10 metabolites. You can see here acid, again, tryptophan, glutamine, succinic acid. It was also contained in previous panels, and also amount of smoking. And in both populations, this test performed very well with above 90% in the ROC curves. It also performed well against controls with non-cancerous lung diseases, so it's good. And also it only required only 40 microliters of plasma, which is making this test fast, minimally invasive, and clinical scalable. We expect with this test to reduce the prices for lung cancer screening in Canada by a factor of 13, reduce a time for the test, 5 minutes per sample. We can do it in 5 minutes per sample. We could increase the performance and to get lower false positive rates, which is one of the disadvantages of the current approved test, which is LDCT. And also we expect to save lives. In Canadians, 15,000 Canadians yearly we can save with implementation of this test. But again, we are in the initial phase, and what we need is larger studies to validate these studies. So the general lessons we could draw about all the examples we have mentioned here is that guidelines move slowly, 10 to 15 years behind science, because even when the evidence is clear, these guidelines changes require systematic reviews. This is one thing we are trying to do in the case of prostate cancer and lung cancer, to get all the evidence and meta-analysis about the participation of these metabolites in the real screening test. Also, a consensus for multiple regulatory bodies, alignment with global regulatory agencies, and cost-effectiveness analysis. And the best example we can mention is ApoB. ApoB, we know that now there are standardized, automated, accurate, and inexpensive methods to measure ApoB, which are currently available in some countries in Europe and also in the United States. However, the European Society of Cardiology and the European Atherosclerosis Society retains the low density lipoprotein C as the primary measure of cardiovascular risk, even when the analytical performance of ApoB is superior to the measurement or calculation of LDL-C for cardiovascular risk, because they are based on present universal acceptance of LDL-C in medical textbooks, and these clinical guidelines have not been updated in more than 25 years. So it takes time, it's a large and long journey, and time consuming, and of course, not all the biomarkers will be successful in this road. This is a collective work, and we can say that we need quantitative data. It's mandatory to have quantitative data. Having access to this absolute concentration will represent a gold standard for these biomarkers approvals. We need harmonization, standardization protocols, certified instruments. We know that in the case of metabolomics, this is really variable, the instrument status and the instrument performance between different laboratories. So we need to harmonize and standardize the conditions of most of the instruments. We need more certified laboratories and quality management systems implemented in these certified laboratories. Also, we need grants to make possible large studies, global trials, and validation cohorts. And of course, we need to demonstrate a clear impact in clinical outcome decisions. If we cannot demonstrate the clinical outcomes of a selection of these biomarkers for a particular disease, we will fail in the process. So this is what I wanted to share with you. I would like to thank my university in Mexico, University Autonomous of Zacatecas, the Metabolomics Innovation Center, and the University of Alberta here in Canada, the National Council of Science and Technology, Mexican Institute of Social Security, and some particular collaborators, Dr. David Wisher, Dr. Juan José Oropeza, almost Dr. my PhD student, David Garcia Alejandro, and all the staff from TIMIC Wisher Note. Thank you so much for your attention. I'm able to take some questions.
Thank you, Dr. Yamilé. It was an amazing talk. We have a couple of questions here in the chat. We encourage you to leave more questions if you like. So I'm gonna read the first one. So, have you observed any additional lipid conversions, such as infingomelin to ceramide or any other, when samples are left at room temperature?
I did not measure that. I was focused on phosphatidylcholines and lysophosphatidylcholines, but we know that these conversions exist. We know that oxylipins are highly dependent on the variation and fluctuation of temperatures. So for sure, there are so many papers that have demonstrated the conversion between different species of lipids. I was just only focused on this because it was just to track some changes in these samples and to know or to confirm myself that I couldn't use those samples for anything else in the lab. But it was not for purpose of investigation or for research. But I can say that, these changes probably happen for most of the other lipid species and also for some other metabolites, not only lipids.
There's only so much you can monitor every time, right?
Exactly. Yeah. Because I said initially, it was only a couple of hours, maybe 4 hours. The temperature here is not so hot. Probably they were not affected. But anyway, I tested, of course, the samples were affected by temperature. Even with 30 minutes, it happened.
Nice. I'm gonna move to the next question because I'm also very interested about this one. It's asking, do you think AI could improve the integration of metabolomics to the clinical setting?
Yeah, for sure. This is our world with AI. So clinical settings is not an exception. And I attended last month in Montreal the workshop, the Congress for Mass Spectrometry for Clinical Laboratories. And in this Congress, some researchers presented workflows and guidelines to implement artificial intelligence with the clinical settings. So now it's everything on the papers, and guidelines are being established, because of course, it has challenges. We cannot use it with a prior knowledge and a prior consultation. So that's why, as biomarkers, they also require guidelines and proper and standardized protocols to implementing.
Awesome. I'm gonna move to the next question. Says, thanks for the great talk. How many metabolite-based biomarkers are currently FDA approved? Where could one look up this information?
I have found this information looking in the FDA and papers and some reports we have in the lab. We know that in the case of metabolomics, we can say at least 300 metabolites. But of course, there are some non-metabolites, for example, as I mentioned, glucose, urea, lactic acid, those that are in the clinical practice, in the routine clinical practice. But in the case of metabolomics, the total amount could be around 300, including the metabolites in the newborn screening, because there are so many carnitines, acylcarnitines, amino acids, and organic acids in this test. And as I mentioned in the webinar, this is the gold standard of success in clinical metabolomics. Most of the metabolites are included in this wide screening.
Nice. I'll move to the next one, which is asking, in the case of the lung cancer panel, what is the difference between smoking and just using the metabolite panel?
When you use smoking, the performance of a panel improve, and you know that smoking is one factor predisposing lung cancer. So if you include this characteristic together with the metabolites, the performance improve in specificity and sensitivity. You can see the paper. There is one original paper published in 2020 in the journal Metabolites. And this is the explanation and comparison when you include, and this is one thing we normally do in our experiments. For example, in our first papers with COVID, we tried the metabolites alone or in combination with clinical characteristics like cough, some demographic factors as age and sex, and we compare, and obviously it improves. As I mentioned, biomarkers is not only one single molecule, but a combination of different molecules that all together interact to make the scenario more clear, but also the specific contents of the disease. If you include specific conditions, you can get the biomarker more specific to your condition. For example, quinoline tryptophan, it's present in most of inflammatory conditions. But if you combine with other metabolites and the specific characteristic of your particular interest disease, it will become more specific and will get more performance than isolated in a single panel.
I'm gonna move to the next question. It says, amazing talk. You explained that most of the panels have a long time to be approved. How could we speed this up?
Sorry?
It says, you explained that most of the panels have a long time to be approved. How could we speed this up?
As I mentioned, we need first clear criteria to select cases and control in the initial discovery phase. Then once we do this, we need validation with a large population. This is a bottleneck because usually we don't have resources to run these large-scale studies. That's why most of us we are doing pilot studies, accumulating data. All the metabolomics papers are inflated with putative biomarkers. However, to get the approval, we need validation in large cohort studies and also immediate changes in clinical decisions. If this panel is informative to physicians that they need to do one test or one treatment or stop one treatment, this biomarker will be successful. We need money to speed this process. We need collaboration. We need involvement of agencies, regulatory agencies. But internally in our lab, what we can do, at least by the moment, is trying to implement quality management systems, because it will guarantee that you will not fail in the future with validation stage, for example. Some of the ROC curve fail because probably they were not measured correctly, probably, who knows? And you are optimistic about this ROC curve, and then when you plan and you invest money and resources and people and patients in larger studies, it will fail because probably you did not correct at early stage some pre-analytical factor or some errors, and you will fail. So the first thing is trying to implement slowly but progressively quality management systems in the labs.
We have a next question asking, when it comes to studying different metabotypes, how granular should the groupings be? And what are the best practices to establish that granularity in different studies?
Well, for metabotypes, it's complex. And we were analyzing recently one study trying to build metabotypes for cognition decline. And even using 1,000 individuals, sometimes it's hard to see any defined metabotypes because we have so many confounding factors. Diet, because most of the metabolome is influenced by diet. So it's complex. For example, for some alterations like cognitive decline, which depends also from gut microbiota, it's totally influenced by diet, by exposomes, but nutritional factors. So in this case, you need a large amount of people. There is no defined sample size for this. You can try. In some cases, depending on your condition, it's more clear or not. You can use some advanced statistic methods, unsupervised methods, machine learning tools to try to explore and to get the clusters, but it's not easy. It's not easy. And most of the researchers that have success on this have used cumulative data or meta-analysis data with large studies, thousands of persons.
So I'm gonna move to the last question. It says, thank you for this very interesting presentation. Sample preparation will influence the detection of metabolites. Do you use one standardized protocol for each sample type? For example, plasma, dried blood spots, serum. If you don't know the biomarker, you don't know which protocol is the best in the beginning?
Yeah, sample preparation for sure is critical, and it depends on the method you will be doing. For example, for on-target metabolomics, we prefer just only protein precipitation and trying to get most of the metabolites in a single phase because the intention of on-target is to cover most of the metabolome. So you will not do specific phases or collection or partitioning of the samples because you will split the metabolome. But in the case of a target, if you want to analyze polar metabolites or more non-polar metabolites, you can choose different strategies. In my case, as we analyze metabolites with target method, but these target are comprehensive method, we use the same as in on target. Just precipitation with organic solvents, for example, methanol, and then we can collect all the supernatant with most of the metabolites, including lipids, in a single phase. For example, just to avoid partitioning of metabolites according to the organic phase. And but this is in the case of target methods that are comprehensive methods. If you know the nature of the biomarker or the metabolite that you want to analyze, you need to know about the physicochemical properties and the solubility in different solvents. And based on this criteria, you can select the conditions properly to get isolation of these kind of molecules. If not, if you're interested in more comprehensive analysis, the best thing is to try to not influence so much in the extraction. And of course, in the case of urine, it's easier because it's supposed that urine in normal conditions does not contain proteins. So you can simply dilute the urine, and in this way you can analyze it. Dried blood spot. There are so many papers reflecting standardized protocols to analyze dried blood spot samples because it depends on the stability of metabolites and the conditions of storage. Some papers recommend the storage at minus 80 with desiccators just to prevent the changes in some metabolites.
We have a final comment from Monica Calla saying, "Thank you, Yamilé, for the excellent talk. It provided a clear overview of the potential of metabolomics in clinical practice, particularly the regulatory aspects that are needed, including the use of internal standards. I'm looking forward to seeing these developments implemented in Colombia and Latin in the near future."
Yeah, for sure. Yeah, we are making Monica Calla.
Can I ask you one final question, Yamilé? Because I'm not from the field of clinical metabolomics, but I was very interested when you showed about the database and the biobanks at the beginning of your talk. And, like I'm based in Europe now, and there are so many publications about this databases that are from Europe, the UK, or North America, but I don't see that much for South America or even like Latin America. Do you think like even people that are based in other countries could use that database in their research, or like how do you see the difference between like North and South Hemisphere pretty much?
No, I think, yeah, we can use it. And what has been done in human methylome database is try to collect diverse data, not only from target studies, for on-target studies, and from so many papers published elsewhere, no matter if they are from Europe or for North America, for South America. So in this sense, we can collect the data, curate the data, and to establish reference values. Of course, the most appropriate is to have geographic databases. We know that, for example, in the case of Mexican population, it's highly influenced by some comorbidities and some conditions, for example, obesity and hyperlipidemia. So in some cases, what is normal for our populations is a little high for other populations. And what we are doing now, at least is my personal interest, is to create our own database with Mexican patients. And that's why I am collecting information about all my papers, which include not only adult people, but also newborns and pregnant and healthy and unhealthy. And all these values, which are quantitative values, I am compiling, and I am creating reference ranges for different conditions, different sexes, and different ages, populations. It's the ideal.
Yeah, because we are on the development stage of in the case of Mexico, right? So we don't have as much money as Europe in this case. We're on baby steps right now. So, well, thank you, Dr. Yamilé, for your amazing talk, and thank you all for joining us today. We hope to see you again in our next EMN webinar this December. I think it's in the 15th of December.
Okay.
And have a
Yes.
great day, you all.
You too.
Thank
All
you
of
for
you.
coming.
Bye bye. Bye.